Welcome back, everyone, to Talk 10 in my series of short talks on epigenetic science. On October 1st, 1990, a huge scientific undertaking began called the Human Genome Project. If you're enjoying these videos, please like, subscribe, share, and follow. It's very helpful and much appreciated. Thank you. I'm doing an episode on this important project because regardless of whether we've heard about it or not, it's had a large impact on all of our mindsets in terms of how we think about our DNA. Have you heard of the Human Genome Project? The Human Genome Project was a landmark global scientific effort whose signature goal was to generate the first sequence of the human genome. DNA sequencing involves determining the exact order of the bases in DNA, the A's, C's, G's, and T's that make up segments of DNA. A genome is the entire set of genetic instructions found in an organism. Can you imagine knowing all of the letters in our DNA? When I was in graduate school at UCSF, there was a huge excitement among the scientists. The thinking was that once we knew the sequence of all human DNA, we would have the keys to people's health and disease treatment. Today, we're going to look at what science predicted would happen at the completion of the Human Genome Project and what the actual results revealed. My name is Robert Needleman. I'm a molecular biologist and biochemist. Welcome to this episode of Empower My DNA. The sequencing of the human genome involved researchers from 20 separate universities and research centers across the United States, United Kingdom, France, Germany, Japan, and China. The Human Genome Project was envisioned to take 15 years and cost $3 billion. Scientists, politicians, and the media were promising many powerful benefits from knowing the entire sequence of human DNA. In order to understand why, we need to know what science thought our DNA did. Here are some major scientific assumptions going into the Human Genome Project. Assumption number 1, the central dogma. A hypothesis was put forth in 1957 by Dr. Francis Crick, one of the Nobel laureates who discovered the structure of the DNA molecule. It became the foundation of modern biology. It's called the central dogma. The central dogma refers to the transfer of information from DNA to RNA to protein, but it says the information flows in only one direction. Francis Crick gave a lecture at University College London in 1957. He drew a diagram to explain the flow of information from DNA to protein, and he wrote, "The central dogma, once information has got into a protein, it can't get out again." Dr. Bruce Lipton, in his book, The Biology of Belief, clearly explains why the central dogma is not correct. The central dogma's assumption of a one-way flow of information from DNA to RNA to protein is profoundly important. Since proteins represent the physical body, the dogma implies that your physical body and your life experiences cannot send information back and alter the DNA. According to the dogma, DNA controls your life, and you cannot influence your DNA. It's important we examine what scientists believed before the Human Genome Project because all 4 of the big assumptions I'm presenting to you are going to be proven wrong. We can influence our gene expression through our lifestyle choices, and we are in charge of our health. Our DNA does not control us. We direct our DNA. From the central dogma came the idea of the primacy of DNA. The primacy of DNA is the belief that DNA is the primary and most important factor in determining an organism's traits and characteristics. Assumption number 3, one gene makes one protein. In other words, there is a gene for every protein. Here's a publication called the Human Genome Project, written by the head of the project, Dr. Francis Collins. One goal was to identify all of the 50,000 to 100,000 genes contained within the genome. At the time, this is how many different proteins were thought to be in the cell. Assumption 4, Human medical conditions are related to changes in the DNA sequence. Dr. Collins goes on to say, this ambitious genome project can provide scientists with powerful new approaches to understanding the development of diseases and to creating new strategies for their prevention and treatment. Now, let's dive into the Human Genome Project. The project went from 1990 to 2003. The final cost was $2.7 billion. Over 90% of the human genome was mapped and sequenced. Human DNA has 3.2 billion base pairs of DNA. The entire genome was completely sequenced in 2022. Each of us has enough DNA to go to the sun and back more than 300 times. In other words, we have a lot of DNA. It's 93 million miles from the Earth to the sun. The results were very exciting and all over the media and the scientific journals. Here's a New York Times headline, "Genetic Code of Human Life is Cracked by Scientists." That's a powerful statement. The big question is, how many human genes are there? Drum roll, please. Here's an article from Nature, The Status of the Human Gene Catalog. Protein coding genes are estimated to number fewer than 20,000. Wow! We have over 100,000 to millions of proteins and less than 20,000 genes? Even tomatoes have almost 32,000 genes. And only 1 to 2% of our DNA codes for proteins. The DNA Learning Center made a great visual for the number of genes in our DNA. Here are the DNA letters, written to fill up pages of paper. Think of how many letters fit on a single page. Now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell. The entire human genome. There are over 3 billion pairs of letters in our DNA. Only 1 to 2% of the letters code for proteins. From the stack of papers of all the DNA, the protein genes are separated out as single sheets. It's really quite amazing. We have more than 3 billion base pairs of DNA and less than 20,000 protein genes. This is an excellent visual showing how little of our DNA actually codes for proteins. These are mind-blowingly exciting results and quite unexpected to the scientists in the Human Genome Project. They also raise some very big questions. What is the function of the rest of our DNA? How do we make well over 100,000 proteins from 20,000 genes? Is more than 98% of our DNA junk? That's an actual term from science. Here's an article. What is junk DNA and what is it worth? In 1972, the late geneticist Susumu Ono coined the term junk DNA to describe all non-coding sections of a genome. A project called ENCODE, short for Encyclopedia of DNA Elements, began in 2003 and is still active today. ENCODE studies all of the DNA that doesn't code for proteins. In 2012, they published a paper showing that at least 80% of our DNA codes for switches involved with our gene expression. The Washington Post published an article, Junk DNA Concept Debunked by New Analysis of Human Genome. The vast majority of human DNA seems to be involved in maintaining individuals' well-being, a view radically at odds with what biologists have thought for the past 3 decades. A very good book called Junk DNA, A Journey Through the Dark Matter of the Genome, was written by Dr. Nessa Carrey. She gives a helpful analogy regarding the concept of junk DNA. In the analogy, the car factory employees represent the DNA. For every 100 employees, it's quite unlikely that 2 people are building cars, i.e. 1 to 2% of the DNA is protein genes, and 98 people are sitting around doing nothing. i.e. the rest of the DNA. What's more likely, 2 people are building cars and 98 people are doing all the other jobs, including designing cars, parts acquisition, accounting, publicity and sales, cleaning, etc. This is a much more likely scenario for our DNA. 1 to 2% of our DNA is protein genes. The rest of our DNA is part of our gene expression. Let's address our second big question. Assumption 3 was that 1 gene makes 1 protein. We have 20,000 genes that make over 1 million proteins. Science has proven that 1 gene can encode more than 1 protein, even up to thousands of proteins. One of the main ways this is accomplished is through epigenetics. Our epigenetics, our gene expression, influences what proteins are made from our genes, based on our lifestyle choices, our perceptions, and our beliefs. I hope you're starting to see how everything we've been talking about so far in my series is coming together. What about the other big assumptions that were made going into the project? Assumption 1 is what was called the central dogma. The central dogma states that information flows from DNA to RNA to protein, but never back from proteins to DNA. Here's the definition of dogma: a point of view or tenet put forth as authoritative without adequate grounds. Francis Crick, who named the central dogma, published a book in 1988 called What Mad Pursuit. In it, he says, I called this idea the central dogma, applied it to a grand hypothesis that, however plausible, had little direct experimental support. As we've been learning in my last 9 talks, our cells interact with their environment, which generates signals which tells the cell which genes to use. Our proteins most definitely affect how our DNA is expressed. The last assumption we looked at, human medical conditions are related to changes in the DNA sequence. This is an article from Your Genome. What are single gene disorders? Individually, single gene disorders are each very rare, but as a whole, they affect about 1% of the population. Less than 1% of diseases are caused by a single gene mutation. Dr. Jean-Pierre Issa was interviewed for a Nova series, Ghost in Your Genes. There is actually a lot more epigenetic change than genetic change in the majority of cancers. The European Journal of Internal Medicine had a very important article, How Epigenetics Impacts on Human Diseases. Recent studies suggest epigenetics may be critical in various diseases, from cardiovascular disease and cancer to neurodevelopmental and neurodegenerative disorders. Epigenetic modifications are potentially reversible and could provide new therapeutic avenues for treating these diseases using epigenetic modulators. The 2024 Nobel Prize in Physiology or Medicine was given to 2 scientists, Dr. Ambrose and Dr. Rufkin. They study the DNA that doesn't code for proteins. And they got a Nobel Prize for the discovery of microRNA and its role in post-transcriptional gene regulation. This is a form of, you guessed it, epigenetics. The Human Genome Project was a great achievement in many ways, and to me, one of the biggest results was showing us quite definitively that our DNA is not our destiny. We are not victims to our DNA. We are in charge of our life by how we use our DNA by affecting our gene expression. One of the greatest professors I had at UCSF, in whose lab I had the privilege of working, Nobel laureate and former head of the NIH, Dr. Harold Varmus, said this: DNA taken from different individuals, especially from people of different ethnicities or originating in different parts of the world, is 99.9% identical. I came up with an analogy that may help us understand epigenetics even more. The English language is written with 26 letters. The 26 letters are what is called the Latin alphabet. Do you know how many languages are written with the Latin alphabet? Over 1,000. Our genes are like the letters in the alphabet. They can be expressed in many languages, including health and longevity, or illness and aging, all based on our lifestyle and mindset. It's up to us. I hope this analogy helps us think about the massive potential of epigenetics. We have the power to express our DNA any way we choose, based on how we live and how we think. We can make this empowering statement: my body uses my DNA for my perfect health and longevity. Thank you very much for joining me today. I hope to see you on my next short epigenetics talk. Have a wonderful, healthy, happy day.